To first learn about epistasis, we need a background. And so a gene is a unit of heredity that codes for a trait, and an allele is a different form of a gene, and it comes in dominant and recessive types, and the dominant will mask the expression of the recessive type. A genotype is the genes your body contains, and a phenotype is the physical expression of those genes, such as eye color. And finally, for a definition of epistasis, is when the expression of one allele is masked by the expression of another allele. And so it is required that two sets of alleles or two loci will code for the same trait for epistasis. And there are many types of epistasis, but for this video, we'll focus on single dominant epistasis. Okay, so as a reference against epistasis, we will have normal inheritance. And so here is a dihybrid cross, Punnett square, meaning that these two parents are heterozygous at these two loci for the one trait. And so that will mean that in these almonds are just representations of the offspring. So that means that these first 9 that I'm pulling out right here are for this one genotype that will represent one phenotype. And these bars just mean that it could be dominant or recessive. It wouldn't change the phenotype. And so these 3 would then be this genotype and singular phenotype. And these 3 would be this genotype and phenotype. And then this last one would be this genotype and phenotype. So that means that there are 4 genotypes possible for 4 phenotypes. Okay, so here we have a specific epistasis form of single dominant epistasis where one allele loci will mask the other set of alleles. So if these two allele loci will be for the same trait, if one dominant A allele is present, then these two genotype forms will be the same phenotype. And so that will be represented in this pathway of enzymes. And so developing through this trait of hungry hippo color, which will be these cups, white, the enzyme will transform it from white to purple if no big A allele is present. But if there is a big A allele present, it will inhibit this enzyme and it will stay white. So we'll see that instead of 9 being the first genotype or a phenotype, there will now be 12 because that big A allele will inhibit that first enzyme from acting. So now there will be 12 white. And if there's no big A allele, we'll have homozygous recessive, and it will be able to go to this purple intermediate here. But now we have the B loci. We'll have the enzyme 2 will be allowed to go to green if there is one big B allele. So that means that these 3 will be green. And the last one, if there are both homozygous recessive for A and B, it will stay at this purple intermediate. So there's only one here. And that is from this Punnett square represented. So here we have another cross where the parents have different genotypes. So that will mean that the offspring will have different phenotypes this last time. So it's the same situation where there's a single dominant epistasis for the A dominant allele. And so in this situation, we will have 8 that are white, and then the rest 8 will be green.